Hello everyone, this is Thelma, Assistant Professor, Department of Zoology, Fatima College. In this video, you will be learning about the cell envelope of a prokaryotic cell and its function. Prokaryotes are the smallest forms of life that can live independently. They are considered to be the first life on Earth. The domain prokaryote consists of bacteria and archaea. Bacteria are unicellular organisms that do not have a definite nucleus and membrane-bound organelles. They are the simplest of most primitive cells. Both bacteria and archaea share similar features, but they are distinct forms of. Some of the major cell organs of the prokaryotic cell is as follows. The first one is the cell envelope, which is composed of glycocalyx, cell wall and cell membrane. And the second component is cytoplasm, which is made of mesosomes, chromatophores, ribosomes, inclusion bodies and plasmids. And the third one is the nucleoid, which is the genetic material of a prokaryotic cell. And the last one is the appendages, which includes flagellum, pili and fimbriae. Cell envelope. This is the protective covering of a bacterial cell, and this basically has three layers: the outermost glycocalyx, middle cell wall and the innermost cell membrane or the plasma membrane. The glycocalyx is not essential for the survival of the bacteria, but in certain circumstances it helps the bacteria in many ways. First, it protects the cell from desiccation by preventing the loss of water and nutrients, and it protects the cell from toxins and phagocytes. Phagocytes are a type of white blood cells that uses phagocytosis mechanism to engulf the bacteria and to protect the body. They bind to the pathogens and internalize them in a phagosome, which acidifies and fuses with the lysosomes in order to destroy the contents. And finally, it helps the bacteria to adhere to the environment or other bacteria and grow as biofilms. The glycocalyx is chemically composed of polysaccharides with or without proteins. When glycocalyx is thick and tough, it is called as capsule, and when it is going to form a loose sheath around the cell, it is called as a slime layer. The next component of the cell envelope is the cell wall. Cell wall is the rigid middle layer of cell envelope that provides shape to the cells, and also it prevents the bacterium from osmotic bursting in a hypotonic solution. The cell wall is usually composed of peptidoglycan, and this is composed of N-acetyl muramic acid and N-acetyl glucosamine, which in short we call it as NAM and NAG. So these two components, they are linked covalently. Based on the nature of cell wall, bacteria can be broadly classified into gram-positive and gram-negative bacteria. Gram-positive bacteria, they have a thick peptidoglycan layer with a narrow periplasmic space, whereas gram-positive bacteria are more permeable but less susceptible to lysis. On the other hand, gram-negative bacteria have a thin layer of peptidoglycan with a narrow periplasmic space. Gram-negative bacteria are less permeable but more susceptible to lysis. The next component of the cell envelope is the cell membrane. It is the innermost layer of the cell envelope and it is semi-permeable in nature. It retains the cytoplasm and also it prevents loss of essential components through leakage, and also it aids in the movement of nutrients, waste and secretion across the membrane. So finally, coming to the summary, the ultrastructure of a prokaryotic cell wall is mainly made of cell envelope, which is composed of glycocalyx, and this is the sticky layer of polysaccharides that protects the cell against desiccation and attaches to its surfaces, and also it helps evade immune cell attack. The glycocalyx can be thick and tightly bound to the cell, and this is called as capsule, or it can be thinner and loosely bound, which is called as a slime layer. And the cell wall, which provides a structure and protects against cell lysis. Based on the cell nature of the cell wall, bacteria can be broadly classified into gram-positive bacteria, which have a thick wall of peptidoglycan, and a gram-negative bacteria, which have a thin layer of peptidoglycan. And final one is the cell membrane, which represents a permeability barrier, and it acts as a site of transfer of nutrients and metabolites into and outside of the cell. These are some of the references that I have used in this PPT. Thank you.